Welcome to the Synthetic Neurobiology group. Since you can't visit us in person, we did our best to bring the lab to you by having students talk about the active projects that are going on in the lab. Enjoy!
Our lab is using zebrafish as a model organism to study how the entire brain works together to generate behaviors. Zebrafish larvae is ideal for its modest brain size with around 10 to the fifth number of neurons. To understand the spatial organization of cell types, we apply expansion in situ sequencing, which allows molecular profiling of individual cells. To further investigate how different cells coordinate to carry out functions, we use expansion microscopy to map connectivity across the whole brain. Zebrafish have the unique advantage of having a transparent brain at the larval stage, which facilitates imaging. We develop novel microscopes and genetically encoded probes to capture brain activity with close to single-cell, single-spike resolution. We hope to understand complex behaviors like decision-making under different affective states. We hope that zebrafish can help us understand an entire vertebrate brain.
So this is the Boyden Lab cell culture rooms. We have two biological safety cabinets, which, can anyone guess what the blue light is for? It's the UV we keep on in between usage of the hood to keep it sterile. And then we have our incubators. The bottom one is what I make a lot of use of. It's where the neuron culture plates are kept, specifically the brain cells that a lot of the molecular tools I work on get tested in first in these cultured cells before going on for animal experiments or brain slice experiments. And so after the neurons receive the DNA for the molecular tools that are being developed, they come into here to be looked at or tested on our spinning disc confocal microscope. You can see the objective and the plate would go right there. And that's where we'd be able to see the proteins being expressed by the cells after the transfection.
My name is Danielle, and I work on our fourth floor space. So I am monitoring C. elegans activity during decision-making behavior, as well as demoing tools that other people in the lab make in C. elegans. And working on a method for C. elegans neural identification. So other features in the fourth floor space that I use: hoods over here, space for more people to work, some basic all-purpose stuff, incubators for various expansion of C. elegans and other work, big centrifuge, my incubator. I've got several plates of worms of various strains for lots of different experiments. Some more all-use tools, and a microscope that I use for basic worm maintenance, a bunch of other stuff, as well as our other spinning disc confocal that I use for imaging a lot of my RNA fish work.
To wrap things up, hi, I'm Jordan, and as you can see, I am not in the lab because a lot of my work is computational. I'm interested in deep brain stimulation, specifically non-invasive deep brain stimulation methods that might allow us to selectively target deeper structures or circuits in the brain without also recruiting structures and circuits closer to the surface, and ideally without requiring surgery. In particular, I'm interested in temporal interference and interferential brain stimulation method that came out of our lab a few years ago. TI leverages two or more high-frequency electric fields that differ at some beat frequency so that we can stimulate deeper regions of the brain in a spatially selective way, ideally without recruiting other parts of the brain as well. So I'm interested in modeling different temporal interference stimulation setups as it relates to electrical stimulation as well as other approaches, as well as the possibility of multimodality stimulation methods where we combine multiple existing deep brain stimulation methods in a way that allows us to better target the regions that we're interested in. So hopefully you enjoyed this virtual tour, and feel free to reach out to any of us. You can check our lab website for our emails if you're interested in any of the projects that are going on here, or if you just want to chat.